My name is Sheila Brown, and I'm the Executive Director of the Canadian Centre for Ethics in Public Affairs, CCEPA for short, which is a joint initiative of St. Mary's University and the Atlantic School of Theology. And I'm very pleased to welcome you all here this evening. It's great to see such a terrific turnout. Our mission at CCEPA, which is based here in Halifax, is to provide an arena for critical thinking, discussion and research into the current ethical challenges that confront us as a society. We do this in a number of ways. We sponsor public presentations, such as this evening's lecture. We support research, and we assist organizations who are looking to integrate values and ethics into their plans and operations, into their governance. Each year we deliver a series of public lectures where participants can engage in discussion on topics of public importance. On behalf of our Board of Directors and its Chair, Alan Shaw, who is here this evening, Chris Stover, our General Manager, I'd like to thank you for joining us as we conclude this 5-part national series, Trust in the New Sciences. This series has explored the ethical, philosophical, and social implications of the advances in genomics, neurosciences, and nanotechnology. Our speaker tonight on genes, genomics, and human nature is Dr. Evelyn Fox-Keller, Professor Emerita of Science, Technology, and Society at the Massachusetts Institute of Technology. We're absolutely delighted to have Dr. Fox-Keller conclude our series. She's a highly respected, Harvard-educated scholar and the author of many highly regarded publications, including The Century of the Gene. And if you talk to people who've read her work, what everyone says is how articulate she is and how clearly she explains concepts, and she is, as I said, held in very high regard. She's also very well regarded for her feminist contributions. Dr. Fox-Keller has explored the relationship between gender and science, and in particular the way ideologies of gender affect the conduct of science. Her book, Reflections on Gender and Science, has been recognized as an important contribution to feminist theory, and her acclaimed biography of Nobel laureate Barbara McClintock has also advanced our knowledge of women's scientific accomplishments. Dr. Fox-Keller will be introduced more formally in a few moments, but I couldn't resist giving you that short preview. This series is made possible by the collaborative efforts of the Canadian Centre for Ethics in Public Affairs and the Situating Science Knowledge Cluster, funded by the Social Sciences and Humanities Research Council of Canada. The cluster is a 7-year project to promote new ways of bringing together leading scholars studying science and technology from different perspectives and making that work accessible beyond academia. So that's an excellent fit with what we do at CCEPA. Part 5 of this series is also generously supported by Genome Atlantic, by CFAR, the Canadian Institute for Advanced Research, and the Evolution Study Group. We launched our series in this room last October, and we've continued thus far in Vancouver, Toronto, Montreal, and here we are back in Halifax. You can view any of the episodes you missed on our website. Part 1 is already up there. 2 and 3 will be there shortly. And Dr. Fox-Keller's lecture and the immediately preceding one by Dr. Cornelius Bork will go up in due course. Dr. Fox-Keller is also going on to Edmonton to reprise her lecture there. Tonight's presentation is being videotaped. When we open the floor to questions, I'll ask you to go to the microphone in the aisle here for the benefit of a good recording. There's nothing worse than watching a DVD or watching something on a website where you hear the speaker answer a question and you've no idea what the question was. So I'll ask you to please go to the microphone, and that will be beneficial for everybody. You'll also notice around the room there are some evaluation forms. I urge you to complete them at the end of the evening. If you would be so kind as to give us your feedback, it is very helpful to us in the planning of future events. We invite you to join us for a reception afterwards, and of course we'll be checking at the door to make sure that you did your evaluation before we let you into the reception. Now I'll hand things over to Dr. Gordon McEwart of the University of King's College, the director of the Situating Science Knowledge Cluster, to formally introduce our guest this evening. Dr. McEwart.
Evelyn Fox Keller needs no introduction. She just got one, too. Literally, but I found some information talking with her over lunch and last night that you might want to hear about. During the last regime in the United States, the Bush administration, she had her immigration papers ready for moving north. And I'm going to remind her that there's another election coming up, that we might get a piece of her. But she's already partially Canadian. I found out that her family is in part from here, at least from out west, Alberta. Her mother's family, Jewish-Russian immigrants around in the turn in the 20th century, who moved to Calgary, 7 daughters, 7 daughters, one of which her leftist father fell in love with on his way to support the Bolshevik Revolution and got stuck in Canada because they wouldn't let him out. And the whole family established themselves in Calgary and became one of the cultural sort of icons and establishment of the cultural vibrancy that makes up Calgary. But alas, Evelyn Fox Keller is American. She has a BA and her undergraduate degrees in physics from Brandeis University, which she followed with a PhD in physics from Harvard, and burst upon the scenes of anybody. You'd have to be dead not to know Evelyn Fox Keller, and even then, you might know her. Her work burst upon the scene with the analysis of the life of Barbara McClintock and the rather interesting way that Barbara McClintock began to see genes and heredity and the nature of the organism in the work of Feeling for the Organism, published in 1983. And then her sort of, so well-read book, it's almost dog-eared, Reflections on Gender and Science in 1985, where she analyzed science studies, the philosophy of science, psychoanalysis, and a number of other questions in relationship to gender. And then her numerous detailed studies of the philosophy, culture, and history of modern biology, Secrets of Life and Death, Reconfiguring Life, and I think the penultimate book, or the one that's really caught the world by storm, Canada by storm, The Century of the Gene, which was published a few years ago, which she tells me is being ignored somewhat in the United States. Attacked vociferously in the United States, and supported on a bestseller high on Amazon.ca in Canada. Evelyn Fox Keller.
Well, thank you very much for those terrific introductions and for the invitation. It's always a pleasure. This is not my first time to Halifax, but it's a place I love, and I've been here a number of times, and I'm delighted to be back here. So thank you. So my talk tonight, I'm going to talk about what recent biological research has taught us about human nature. But first, I want to begin by asking, what do we mean when we speak of human nature? And I want to try to identify the various assumptions that have attached themselves to that question. Basically, there are two variants of the question, one which puts the stress on the word human, and the other on the word nature. In the first case, the contrast class, that is, what is not human nature, is non-human nature. That is, what it means to be human versus subhuman, animal, etc. What distinguishes the human species from other species? Thus understood, the focus is on the qualities and traits shared by all, or virtually all, humans, including and often concentrating on the ways in which humans tend to think, feel, and act. In other words, here the focus is on human universals and on the differences between these qualities and the universals shared by members of other species. In the second case, the contrast class is what is human but not nature. That traditionally, that means what is acquired by humans after birth, what accrues from nurture, from culture, or just simply what is learned. The focus here is not on human universals, but on human differences. What makes us different from one another? But in both cases, the concept of human nature carries with it two additional assumptions. The first is that whatever the answer, what is natural is simultaneously fundamental and inherent inside. It is essential, inbuilt, inborn, and at least since the 19th century, internal to the physical body of the organism. And by the 20th century, as we know, the locus of the natural contracted even further as it became increasingly confined to the locus of genetic, of genes. The second tacit assumption that often comes with the concept of human nature is that what is natural is more or less fixed, unchangeable. By and large, these two readings of the question, what is human nature, call for different methodologies and might even be said to correspond to different subdisciplines of biology. Nonetheless, at least until recently, there's been a strong tendency to house efforts to answer both variants of the question in the same biological subdiscipline: behavioral genetics, sociobiology, evolutionary psychology. They tend to group together. And to root most such studies, most such efforts in studies of heritability. But these days, talk about human nature has begun to fall under the provenance of genetics and genomics, where it is now being studied with all the available techniques of molecular biology. I will not say much tonight about sociobiology or about measures of heritability in behavioral genetics. For that, I have a book coming out on the mirage of a space between nature and nurture. But instead, tonight, I want to focus on what recent work in genetics and genomics has taught us about human nature, and particularly what it has taught us about what makes us human as distinct from non-human. Curiously, and this is what makes the subject so interesting, the answers that genetics and genomics have been providing to that question point, both conceptually and methodologically, in two very different directions. And I suggest that through this divergence, they illustrate a fundamental tension that has been emerging over the last several decades in biological research, and that may now be coming to a head. The first contribution of recent genetic and genomic analyses, and almost surely the contribution that's most conspicuous in the public arena, is a confirmation and extension of the assumptions that became increasingly prevalent over the course of the 20th century, namely that human nature can best be understood in terms of its genetic underpinning. Indeed, that human nature is written in our genome, or in the totality of our genes. And since 1953, in the nucleotide sequence of our DNA. Indeed, this was the assumption that underlay the mammoth program to sequence the human genome. As Wally Gilbert famously put it in the early days of that project, we will be able to put the sequence of our DNA on a CD and say, this is me. Early drafts of a generic human genome first began to appear about a decade ago, and over the years since, the enthusiasm both for generic sequences and for sequences of individual genomes has continued to grow. As individuals, we want to know about our ancestry, our propensity for particular diseases, personality, cognitive abilities, and now as well, about what is natural about our political orientation, about who we vote for. As members of the human species, just what is the difference between man and ape? Just what is it that makes us by nature a political animal, a social animal, or an animal who speaks? Although I started out by trying to separate these two kinds of questions, one about individual differences and the other about species differences, or species characteristics, they tend to get grouped together in these efforts, and the expectation continues to grow that we can find the answer to both kinds of questions in the sequence of our genome. The cost of sequencing an individual genome is going down very rapidly, and it's expected to reach a mere $1,000 within a decade. Indeed, a number of private companies, like, for example, Navigenics, DecodeMe.com, or 23andMe, have already emerged with offers to members of the general public to sequence parts of their DNA for less than $1,000. And people are buying. Anyone can do it. Just Google personal genomics and you'll see. 2 years ago, Science magazine put out a special issue on what the editors called the last frontier of genetic analysis, the genetics of human behavior. As they wrote, the potent genetic tools available for Drosophila have uncovered many genes that, when deleted, disrupt behaviors, like, for example, mating behavior or circadian rhythms. This, in turn, has allowed dissection of the neural circuits that control essential behaviors. Inevitably, we are led to ask, if geneticists can do this for Drosophila, why not for humans as well? Already, genetic analysis of mice has succeeded in identifying a number of genes that are said to underlie circadian rhythms, and efforts to find genes associated with other behaviors such as anxiety, addiction, learning, and memory, are moving forward rapidly. To be sure, mice are not men, but they're a lot closer to us than Drosophila. And much can be learned about humans by studying homologies between human and mice, or even Drosophila DNA. But for direct genetic analysis, humans present particular challenges. Geneticists are not allowed to do controlled mating experiments, or to freely manipulate human DNA, at least not in whole organisms. Also, human behavior is far more variable than it is, or behavior is far more variable in humans than it is in flies or mice. As a consequence, geneticists who study human behavior have traditionally had to rely on statistical analyses of genetic variation in populations rather than on an experimental analysis of individual lineages. Recently, however, methods of neural imaging have opened up the possibility of observing neural activity in individual brains, and many hope that these techniques can help bridge the gap between experimental and population genetics of behavior. For example, in that same issue of Science, Fowler and Schreiber argue that evidence of a correlation between human genetic variation and variation in political behavior, when combined with studies based on neural imaging, demonstrate the need for a new science of human nature. They recognize that this new science will require comprehending human biology in a socio-political context, but genetics lies at the base of this new science. And they conclude, it will demand that we recognize that genes are the institutions of the human body. They regulate the neurological processes that drive social and political behavior, close quote. Unfortunately, however, efforts to develop a genetics of psychological and political behavior have run into some rather serious stumbling blocks. Despite high expectations and lurid newspaper headlines, we have yet to find the genes for human universals. For example, the genes that make us political animals, that make us social animals, that make us speaking animals. And despite occasional claims to the contrary, we have not done much better at finding the genes for individual characteristics. For example, genes for mathematical genius, shyness, liberal politics, ruthlessness, or adultery. All these continue to elude us. It's not just that these are complicated traits that require the involvement of many genes, each of which contributes only a small part to the total variation. Take, for example, IQ, a trait of long-standing interest to behavioral geneticists and one which has repeatedly been claimed to have high heritability. That is, approximately 50% of the variance in human IQ is said to be due to genetic variance. No one claims to know all the genes responsible for IQ, but a few genetic markers have been identified as playing particularly important roles in verbal and mathematical abilities. And so, in an effort to bridge the gap between the statistical studies and direct genetic analyses, the question was asked, how much of the variation in IQ of a random population could be attributed to variation in these various genes that had been identified? The answer was a great disappointment to students of heritability. Constance Holden puts it like this: if the IQs of the population in question ranged from 80 to 130 points, the biggest gene effect the researchers could find would account for less than 1/4 of one IQ point. And she sums up the state of affairs by noting that it seems to be much easier to identify genes for disabilities than for abilities. In other words, geneticists have been able to identify many genes of a kind that, when they are present in mutant form, appear to be correlated with mental defects, but they have had no such luck in identifying genes for positive mental functions. Holden concludes that genes for particular skills, like mathematics, just, quote, just don't seem to exist, close quote. So what is going on? I want to argue that the stumbling blocks that confront the prospects of a genetic psychology tell us something important about the mission itself, suggesting that the problem is far deeper than the fact that complex traits involve many genes. It tells us something about the nature of genetics and about the limitations of that science. Genetics is, in the first instance, the study of mutations and their effects on phenotypic traits. And it has historically been driven by what I have called the logic of subtraction. In other words, by the assumption that it ought to be possible to understand the genetic basis or cause of a trait, that is, identifying the genes for that trait by identifying all the mutations that can cause the trait to go awry. In other words, a bit like trying to understand human vision by studying all the ways in which a person can go blind. Trouble was already in the air when the first drafts of the human genome sequence began to appear around 2000, and the surprising news came out that the human genome has only, the claim then was that it has only 30 to 35,000 genes, and genes are used here in the sense of protein coding sequences, roughly the same number as in the mouse, and only a little over twice the number of genes in the lowly fruit fly. Since then, estimates have moved steadily downward, today hovering around 20,000 genes. Not only was this number far below anything that had been thought necessary to make an organism as complex as we are, but also it left the troubling question, if humans and mice have the same number of genes, and many of these genes are in fact the same genes, just what is it that accounts for the difference between a man and a mouse? Since 2000, the problem has steadily exacerbated. Not only has the number of genes gone down, but so too has the proportion of DNA devoted to coding for proteins. Current estimates are that only 1 to 2% of human DNA consists of protein coding sequences. And the obvious question arises, what is the rest of the DNA doing? In the past, when non-coding DNA was recognized to exist, whatever part of the DNA that was recognized as non-coding was dismissed as junk, leaving aside the question of why it persists. But now geneticists are beginning to learn of enormously complex biological functions these non-coding sequences are involved in. In addition to being transmitted faithfully through the generations, most of the non-coding DNA is also transcribed into biologically active RNA sequences that seem to play crucial roles in the regulation of individual development. Daily, we are discovering new and extraordinarily ingenious ways in which so-called junk DNA sequences participate in the mammoth projects of regulating the spatially and temporally specific transcription of DNA, the construction and translation of messenger RNA, and the positioning, the conformation, and the activity of the resulting proteins. Early concepts of the gene were predicated on the assumption of a relatively simple transformation from genotype to phenotype, one gene, one trait. You could trace the causal chain from a gene to the trait. But now we're beginning to understand just how enormously complex that process is and how demanding it is of ongoing fine-tuning and regulation. This in itself need not pose any challenge to the expectation of rooting human nature in the genome. It need not even disappoint Wally Gilbert's expectation that he will be able to recognize himself in the sequence of his DNA recorded on the CD. But it does require us to recognize and remember that the genome, that is the full complement of an individual's DNA, is far more than his or her complement of genes, however that term is defined. Indeed, Erica Check speculates that it's the junk that makes us human. One might say that all this recognition means is that we should stop talking about genes and talk instead about DNA. That would be good, and it's not nothing. But we have not done so, and so we continue to talk about genes for this and genes for that, and doing so does pose a problem. In particular, it encourages the assumption, inherited from the earliest days of classical genetics, that the genome is simply a string of genes, and the implication that identifying a mutation means one has identified a gene. In other words, it encourages the persistence of the belief that locating mutations that cause disabilities will tell us something significant about what causes the development of abilities. Indeed, it is just this belief that in turn encourages us to collapse the two questions about human nature that I tried to distinguish at the beginning of my talk. It encourages us to conflate the question of what makes us different from one another, what, for example, causes mental retardation in some, with the question of what makes us who we are, what gives us the mental ability most of us have, what makes us human. I suggest that a primary source of this confusion can be found in our failure to adequately distinguish mutations from genes. This is a surprise because it's a simple distinction, right? We know what a gene is, or we think we know what a gene is, and we can say what a mutation is. Why do they get collapsed? Well, the habit of talking about genes and mutation as if they were the same goes back to the early days of genetics and persists to this very day. Thus, for example, we routinely overlook the fact that the map that those early geneticists constructed was not a gene map, which is what we call it, and what it was called in those days, but a map of mutations. Ever since, both biologists and philosophers have become accustomed to talking about genes as difference makers, while at the same time thinking of genes as trait makers. In point of fact, however, what makes a difference in a trait is not a gene, but a mutation. And the question of what makes a trait, of the causes of a particular developmental outcome, is left unanswered by simple studies of mutations. Now, don't get me wrong. I'm not knocking genetics, and I'm not suggesting that it's not useful to study mutations. Far from it. Efforts, for example, efforts to correlate anomalies in the DNA sequence with particular diseases have, at least in the case of some diseases, have clearly been of immense value for diagnoses and for prediction. And someday they may even prove useful for treatment. But it is important to understand that such correlations do not, by themselves, indeed cannot, say anything about genes for those diseases, and for two reasons. First, just as diseases are not traits but defects, departures from a norm, so too anomalies are not genes but mutations. And second, as I've already said, because most of these anomalies in the DNA are located not in protein coding sequences, but are rather far more likely to be located in non-coding sequences involved in regulation. My claim that diseases are not traits, but rather trait differences will surprise many of you, because we have become so accustomed to a language that treats disease as a property of an individual. We commonly speak of an individual as having a disease, much as he or she might have brown eyes. But in fact, and has long been understood by many writers, disease is a state that exists only in relation to another state that has already been established as normal. In his inquiry into the scientific rules for distinguishing the normal from the pathological, written more than 100 years ago, Emil Durkheim stressed that, quote, a trait can only be characterized as pathological in relation to a given species, unquote. In other words, in relation to a standard of normality or state of health, which is itself inextricably confounded with the norm of that species. He goes on, one cannot even conceive without contradiction a species that could by itself and in virtue of its fundamental constitution be irremediably sick. The species is the norm par excellence, and accordingly can harbor within itself nothing of the abnormal. So, to repeat, studies of mutations may tell us a lot about the genetic basis of abnormalities, even while telling us little about the genetic basis of normality. In the same vein, mutation studies can tell us a great deal about the genetic differences that contribute to human diversity, but they cannot tell us what makes us what we are, either as individuals or as members of a species. They cannot give us human nature in either understanding of that term. For understanding how genes or DNA contribute to our abilities, to our positive characteristics, we have to look elsewhere. In particular, we have to look to studies of the dynamics of developmental processes. I will talk about what studies of this kind have taught us about human nature in a bit, but first, I want to say a bit more about the question of what we have learned from genetics and genomics. Recognition of the difference between genes and DNA, on the one hand, and between genes and mutations on the other, was an important byproduct of the successes of the Human Genome Project. But it is not the only way in which we have benefited from all the new information genomics has given us. Far from it. I have argued, in the Century of the Gene, I argue that the greatest gift the Human Genome Project gave us was humility. By spelling out the sequence of human or any other organism's DNA, the Human Genome Project clearly demonstrated that our DNA sequence, as rich a resource as that is not by itself sufficient to make an organism, much less a human organism. The gap between promise and delivery that the realization of that project made visible made also starkly clear the need to focus on the system in which DNA is embedded, for it is the system that gives meaning to the sequences of nucleotides. Genotype, the sequence of DNA, does not spontaneously transform into phenotype. This crucial transformation is not a process that is itself written into the DNA. Rather, it is a process orchestrated by the system of which DNA is a part. By itself, DNA sequence is without meaning. Even those sequences we refer to as protein coding sequences or genes become sequences that code for proteins only when integrated into the cellular system. Naked DNA by itself means and does nothing. In this sense, we must remind ourselves that sequences of DNA are not, by themselves, are not genes. They become genes only within a biological system that gives them that meaning. The genetic information that is claimed to be housed in the DNA is so housed not only by the cell, but also only within the context of that cell. The successes of the Human Genome Project have also had indirect benefits. For one, the techniques and the data banks it provided have helped us begin to recognize sources of biological information that are housed in other parts of the cell, as well as in the environment on which the life of the cell or organism depends. For the cell, the DNA is not so much a master molecule as a resource. Dennis Noble has likened it to a musical score. Making use of the capacities built into its own internal organization, the cell can put this resource to extraordinarily ingenious uses, and that go far beyond the use of sequences to make proteins. The organization of cells, tissues, and organs makes possible all kinds of other ways, and I don't have time to go into that, to use this resource. I have still to speak about the organization of organisms in their environment and the important ways in which this organization too contributes to human development and the construction of the human phenotype, that is, to what we mean by human nature. Traditionally, talk about human nature has treated environmental factors as separable from hereditary factors, as in genes versus environment, thereby not only ignoring the critical role of cellular components that are not directly inherited, that is to say, the environment internal to the cell but not in the DNA sequence, but perhaps more importantly, treating what we think of as the environment as if it could be defined independently of what we think of as the organism. In fact, we have learned that organisms and environment co-construct each other in ways that defy clear separation. And this is true of internal as well as external environments. Consider, for example, the fact that there are at least 10 times as many bacteria as human cells in the human body. These bacteria are neither external to nor independent of the human organism. They contribute crucially to our survival and maintenance, just as we contribute to theirs, in ways and in all sorts of ways that we're just beginning to understand. But already it is clear that they are a part of what it means to be a human being. In other words, if we want to talk about the genotypic contribution to our phenotype, we have to consider the contributions not only of our human DNA, but also of the bacterial DNA that resides inside our bodies. Furthermore, environmental contributions to our phenotype, bacterial or otherwise, go far beyond whatever other DNA can be identified. In short, our phenotype depends not only on our DNA and the cellular machinery that processes this DNA, but also on both the internal and external environment in which we live. Or rather on both the external environment in which we live and the internal environment that lives inside us. Clearly then, an understanding of what makes us human requires more than an understanding of our genome. It requires as well an understanding of the entire ecosystem that defines our phenotype. Finally, a word about plasticity, a notion of particular importance if we're to understand the role of environment in human development. I referred earlier to a second assumption that seems to have attached itself to the idea of human nature, namely the assumption of the fixity of what it is that is given by nature. One challenge to this assumption was raised by geneticists as soon as the idea of genetic manipulation took hold. But another and more serious challenge comes from more recent biological research demonstrating the importance of plasticity and adaptability in the development of organisms, and especially in the development of humans. And even more especially in the development of human mental capacities. Increasingly, we seem to be coming to the view that what most critically distinguishes humans from other animals is neither our DNA or the particular traits that we have, but our unusual capacity to adapt to and learn from different kinds of environmental inputs. One might say that it is our nature to be nurturable, to be cognitively pliable. The idea of neural plasticity was first proposed in 1890 by William James, but for the better part of a century it lay dormant. Today, neural plasticity is the hottest subject of neuroscience. Contemporary research suggests not only that our brains, all areas of our brain, are to a greater or lesser degree plastic throughout our lives, but also that thinking, learning, and acting actually change both the brain's physical structure and the functional organization from top to bottom. To be sure, the plasticity of our brains has to come from somewhere, and it does seem, indeed, to be inborn. Furthermore, it seems to be a universal property of humans. We are, virtually all of us, born with the capacity to learn language, just as we are born with the capacity, indeed, the need to interact with others, to evoke responses from our caretakers, to mimic their expressions and actions. In these ways, we begin to construct our human environment just as and in concurrence with the ways in which the environment constructs us. But even so, I don't know of many efforts to identify genes for plasticity per se. Now, some of you may have seen a recent report in which researchers at MIT identified a protein called ARC that appears to control neurons' the ability of neurons to strengthen or weaken their synapses by regulating the number of neurotransmitter receptors on their surfaces. I suppose one might refer to the gene for this protein that plays such a crucial role in the neuronal plasticity. One might refer to that gene as a gene for plasticity, but to me that would seem to miss a major point, namely the importance to plasticity not so much of the gene itself, but of the level of gene expression. In a similar vein, there are also a number of scientists searching for language genes, but still, it seems to me that the very concept of plasticity suggests that we look for its roots not in the parts of which we are made, but in their dynamic organization. All this talk about organization might be said to hark back to an earlier time, indeed to the very beginning of what is called the science of biology, a time when it was widely assumed that the essence of life was to be found in its organization rather than in its material components. It is talk about organization is also clearly at odds with 20th century talk about genetic primacy and genetic reductionism or genetic determinism. But as the genomics of the 20th century begins to give way to the systems biology of the 21st century, to studies of the relation between parts and wholes, the notion of organization and its centrality in biology has taken on a new life, as it were. One might say that we are witnessing a sea change in biology in which confidence that an organism can be reduced to its genetics parts is giving way to the recognition that parts are not enough, that biological explanation of human nature, or indeed of anything else, requires attention to the systemic organization of these parts. Indeed, many see a further shift in which the organization of the system has usurped the position of primacy, and the genes, or even the DNA, have become relegated to the position of components or parts that depend on their organization for their meaning. To sum up, it would appear that current biological research is giving us a rather different view of human nature than that with which the Human Genome Project began. The new view is in fact in rather close sympathy with one that has been gaining ground in psychology, in cognitive neuroscience, and in developmental theory. I can do no better than quote from your own native son, Merlin Donald. And Donald describes this alternative view of human nature as based on a different set of assumptions. And the first such assumption is that human nature has been characterized by its flexibility, not its rigidity. And this is Merlin Donald I'm reading from. This is due largely to the overdevelopment of conscious processing and those parts of the brain that support it. The second is that human beings, as a species, have evolved a completely novel way of carrying out cognitive activity, distributed cognitive cultural networks. The human mind has evolved a symbiosis that links brain development to cognitive networks that can change radically. Critical mental capabilities, such as language and symbol-based thinking, as in mathematics, are made possible only by evolving distributed systems. Culture itself has network properties not found in individual brains. The individual mind is thus a hybrid product, partly organismic in origin and partly ecological. One consequence of this idea is that human nature, this is still Merlin Donald, viewed in the context of evolution, is marked especially by its flexibility, malleability, and capacity for change. The fate of the human mind, and thus of human nature itself, is interlinked with its changing cultures and technologies. We have evolved into the cognitive chameleons of the universe. We have plastic, highly conscious nervous systems whose capacities allow us to adapt rapidly to the intricate cognitive challenges of our changing cognitive ecology. Close quote. Well, a lot of words. What does it mean? What does Donald mean? In particular, what does it say? What is it saying about where human nature comes from and about where it is going? Let me try to translate Donald's argument into my own terms. Almost 400 years ago, John Donne gave us the memorable line, "No man is an island entire of itself." What Donald is saying is that no mind is an island entire of itself. And I am trying to remind us that the same must be said of our bodies and even of our DNA. For a while, the idea that human nature could be located in a fixed essence found a comfortable home in our discovery of the biological importance of DNA, of this enormously complex molecule. But the more we have learned, the more we have come to recognize that the ways in which nucleic acid sequences are deployed by the cell are at least as important as the sequences themselves. And how they are used depends, in turn, not only on the immediate context of the DNA, the proteins in which it is packaged, for example, but also on its larger cellular and extracellular context. Somewhat more surprisingly, the nucleotide sequence is not the only relevant material that is passed from one generation to another. Aspects of its context, for example, the chromatin markings of the DNA, are also transmitted. Indeed, the inheritance of context, and hence also of context-dependent selection pressures, provides one of the primary means by which experience leaves its long-term imprint. When Donald suggests that the individual mind is a hybrid product, partly organismic, by which I take him to be referring to its internal embodiment, and partly ecological, I want to suggest that so too is the biological meaning of the genome, partly structural and partly ecological. Indeed, I would want to say that whatever we mean by human nature, referring now to both body and mind, is necessarily partly structural and partly ecological. Similarly, I need to add that structure and ecology are themselves intertwined. Since the structures of biological organisms are products of evolution, they carry the history of their past experiences, their past physical and chemical ecologies in the structure of their bodies, just as the structure of our brains, as Donald reminds us, carries within it the history of our past cognitive ecologies. Thus, if we want to understand what makes us human and what makes us, say, different from chimpanzees, we have to look at far more than the difference between our generic genome sequences. Estimates of that difference, the difference between human DNA and chimp DNA, vary from 1 to 4%, depending on how one counts, and that is surely not nothing, particularly when the differences occur in regulatory regions of the DNA. But it is also not everything. Whatever physiological differences may have existed when we first branched off from chimps must surely have been exacerbated by the different experiences that original physiological difference made possible. Some of the history of these different experiences will surely find a record in our current DNA, but DNA sequences will not tell the whole story. Over the course of our respective histories, new ways of processing these sequences have also emerged, and these too were passed on, sometimes through the DNA and sometimes epigenetically. Finally, let me return to the question of human natures, that is, to the question of intraspecific variation and the nature of individual humans. Given the complex dynamics of development and evolution that I have sketched here, John Dupre argues that we should expect a great historical and geographical diversity of human natures, close quote. I agree. But Dupre goes on to suggest that we not talk about a single human nature at all and consider only diverse human natural histories, close quote, and here I would demur. I would want to replace his only by also. And I would want to emphasize that the diverse natural histories of human beings living in different cultures and different climes have left their marks not only on individual human natures, but surely also, especially to the extent that, that they have evolved in relative isolation, on the minds and bodies of groups of people. And once again, we need to remind ourselves not to expect the marks of these differences to be confined to the genome. Merlin Donald is also interested in the future of human nature, particularly in the fate of the human mind. And he writes that just as changing cognitive ecologies and technologies, for example, the invention of reading and writing, have shaped the evolution of our brains in the past, and just as more recent technologies like the computer and the internet are changing the structure of our brains today, so too will future changes in our cognitive ecologies and technologies change our brains in the future. I agree here too. But once again, I want to add to Donald's list of cognitive ecologies. For example, consider the impact of more and more refined techniques of genetic engineering. Just because we can't find the gene for mathematical ability doesn't mean that we can't use genetic interventions to induce significant changes, for good or for bad, in our cognitive capacities. But think about it. Science and technology are themselves cultural products. So wouldn't such changes reflect just another, albeit a very different way in which the evolution of our mental capacities depends on culture? Finally, our collective beliefs about the nature of human nature are also part of our cognitive culture, and they too, these beliefs themselves, leave their marks on the development of human traits. Thus, for example, I learned from Dupre that the teaching of economics reduces students' disposition to cooperate. As Dupre writes, hence making economists' typical assumptions about human behavior more nearly true, close quote. In a similar vein, it seems that there is also some evidence that the teaching of evolutionary psychology reinforces sexist attitudes. But culture is not uniform, and the belief systems that people are reared on obviously vary with their particular cultural environment, just as access to genetic therapies will inevitably vary with class. Should we not, therefore, also expect the future development and evolution of our various human natures to also carry marks of these differences, the differences in our belief systems, our class differences? Maybe implications of this sort should make us just a little bit cautious and a little more self-reflective about our own beliefs about human nature than we have tended to be in the past. Thank you.
A Nova Scotian, Dr. Dawson, discovered something that we usually hear as being the first person to put DNA to work in a test tube. He, working with another couple of Nova Scotians, Oswald Avery and Colin MacLeod at Rockefeller and Columbia, New York, he's usually given credit for being the first person to put DNA to work in a test tube. So the story is that an intelligent scientist takes a chemical DNA, sticks it in a test tube with bacteria, and they kind of force the bacteria to change themselves, to be transformed. But that's not what he said at all. What he said, he spent 2 years trying to get them to do something in chemical terms. You put a chemical in, something happens. And what he discovered was the bacteria took their good old time. And for a very short period, they would, in a collective decision, take up the DNA and transform themselves, something that at that time humans couldn't do. We were using X-rays on fruit flies, and most of the fruit flies were being zapped to death. But what I'm wondering is the scientific interpretation of that report of Dawson's was to focus on the fact that humans, or he had, you know, with DNA, the sort of both of them active agents, had forced these stupid bacteria to do their bidding, when in fact what he saw was that bacteria were kind of cleverer than us, because they had the ability to decide when, and in a collective decision, quorum sensing we call it today, to take DNA up and put it into their genomes in their own terms. And this being 1930, and what was in the air at the time was a guy at the same university, Hermann Mueller, and I don't know if you know your genetic history, he was just using radiation basically to try and force genetic changes. Basically a sort of a pre-Hiroshima type style. You know, a lot of losses, and occasionally you got some mutations you could do something with. So what happened? That is 80 years ago. Why did they take one route, ignore the quorum sensing, and focus on kind of a chemical view of DNA as being an active ingredient? As soon as you stuck this chemical in, whatever, you know.
Dawson is the name? I don't know this.
Well, okay.
How do you spell it?
Dawson is a well-known Nova Scotian name. His name is Henry Martin Dawson. Martin Henry Dawson.
D-A-W-S-O-N?
Yeah.
I'm really puzzled by this, because I thought bacterial transformation didn't come until much later than Avery and MacLeod, actually.
No, it came before that. Avery was his boss and was quite opposed to what he was doing. It was sort of against what he'd spent his life on. And very late in life, Avery sort of put a stamp on the chemical determination of DNA, but they could have just as easily went looking for the chemical determination of the chemical that allows various bacteria to communicate with each other and decide, we've got a quorum, let's create a biofilm, let's change ourselves.
Where was this published?
At the GEM, a Journal of Experimental Medicine, Rockefeller's.
Dawson's paper.
Yeah.
But Dawson didn't himself go to Rockefeller.
Yes.
He did?
Yes, and at Columbia. He was the only one who published more than one paper. I mean, up until about the late 40s, most people published one paper on the subject and disappeared, including Avery, etc.
I have to check that out. But there are many examples. Well, I gave one example in my talk, neuroplasticity, proposed by William James in 1890. So there are many suggestions. There are many experiments that just disappear, that are ignored because nobody knows how to fit them in with their current thinking. That's what happened to Barbara McClintock. Happens all the time. But I want to check out the Dawson.
Well, as I say,
very
10 years ago,
interesting.
you would have seen Dawson as a footnote in molecular genetics. He's the guy who puts it into the test tube, then goes on and on and on, sort of a steady progression. Today, if you kind of look him up, you're going to find him he's the first guy to see quorum sensing. So it's the same experiment. It's old data with new eyes.
Yeah.
You briefly mentioned epigenetics. I was wondering if you see the study of epigenetics as a return to the idea of inheritance of acquired characteristics. Like, was that idea correct all along? Like, are we seeing that now?
Well, Eva Jablonka and Marion Lamb, who have been championing epigenetic inheritance for 10 years at least, want to make that argument. They want to claim this as a vindication of Lamarck and of inheritance of acquired characteristics. I don't know what Lamarck meant by inheritance of acquired characteristics. It's not the same. I mean, yes, it's true that as I said, our experience, our environment our histories leave their mark on our bodies, and not just in the sequence of the DNA, in ways that can get transmitted. In that sense, we do inherit, we can, at least for a couple of generations, acquire characteristics. But I doubt that there's much commonality between that formulation and what Lamarck meant.
Thank you.
Thank you for that. Of course, we all know your work and contribution as a feminist scholar. And one of the things I noticed, I was making note as you were speaking, that there was a key word from the feminist scholarship lexicon that was absent from your talk, and that's the word agency. I don't know if I heard it once in the entire discussion. But on the other hand, I think it was implied in a lot of places. It seemed that you were speaking about the agency of organisms and the agency of mind and of cognition and of ecology, and it was widely distributed. Was it conscious to leave the word agency out of it, or maybe you can just talk about the idea of agency in this larger set of connections that you've drawn together for us?
I mean, when feminists talk about agency, they're talking about human agency. And the question of whether using the concept of agency for molecules or for molecular structures is one that puzzles me a great deal. So I don't know how to think about agency in the sort of in molecular terms, and I'm struggling with it. So it's not that I purposely avoided it, but I don't know how to speak of agency in these terms.
You mentioned about disease, about the way that
disease is always in relation to a previously constituted norm. Now, I would like to hear you talk maybe a little bit more about what some of the results of our concentration on genetics and on genetic markers for possible risks of disease mean in the context of our future view of human behavior. And I'm thinking right now about newborn testing, which is happening widely in the United States without any without parents having any sense of being able to consent or not consent to that newborn screening. It's mandatory newborn screening, and when the newborn fails the test, it's not necessarily that doesn't necessarily mean that they're going to have a disease. It just means that they might have a genetic mutation that may or may not cause a disease later on. And so I'd just like to hear you talk a little bit more about that. Those
genetic screening is another example of a technical culture that has an impact on our bodies, and it has direct impact on our genetic futures through selective abortion. There's no question about it. The question about the relation between the significance of the correlation between sequence anomalies and particular diseases is a really tough question. We can identify, we can see the correlation. It's very rare to be an absolute predictor. It's only, in most cases, it's only a statistical predictor, and it's limited to a very small number of quite rare diseases. We really can't do anything of the sort for the more common diseases. So the ethics of genetic screening, I'm no expert on this subject, and I know people who are. So one of the best people to read on this is Diane Paul, but I'm not a knee-jerk opponent. If as an Eastern European Jew, I would want to have prenatal screening for Tay-Sachs syndrome. I would not want to go through that hell. So I don't have any clear-cut, simple response to your question.
And I have a second question that's a totally different question, shifting a totally different gear. I recently read a paper that you wrote on the taking up of technical language of competition and reproduction in its colloquial use. So whereas Darwin used the idea of competition in a very technical sense, then people after Darwin taking that up in a colloquial sense. And I was just wondering if there was anything that you were seeing in the language of genetics that was similar to that.
Yes, absolutely. So, for example, the word heritability has a tremendous amount of confusion has been caused in discussions about nature nurture by the fact that there are two meanings of heritability. One is the common sense meaning of heritability as the capacity to be transmitted through the generations, and the other is a very technical statistical measure. And what one has to do with the other is very unclear. But the use of the same word keeps people from making the distinction and automatically attributing to the technical measures of heritability all kinds of assumptions about genetic transmission. So that's one example. Yes, it's all over.
Thank you very much for your talk. It was fascinating. And I actually had a perfectly reasonable question. I thought it worked out earlier in your talk, and then your final section just completely blew me away. So I'm a bit discombobulated. But I really want to pick up on this question of disease, because you said early in your talk that, disease implies or somehow has ontologically or logically or even epistemologically prior a notion of health. And
of
it's
normality.
That, of normality. Yeah. And it's that normalcy that then allows the disease to be defined. But in fact, if we pick up, for example, Foucault's analysis of the birth of the asylum and mental illness, what we find in his account is that, in fact, it's the defining, the erection of the pathology that allows then for determination of what counts as normalcy. So I think I'm not quite sure how to think it through, but it seems to me particularly important here that it's Foucault on mental illness, not the Foucault part, but the mental illness part, because the question of rationality, when you said early on that, human nature, we are the political animal, we are the social animal, I was waiting for the Aristotelian moment of the zoon, theoretikon, or the animal rationale, that we are the rational animal. And you didn't pursue it at that point, but you came back to it later in your paper when you talked about Donald and how the phrase that was used is distributive cognitive distributive cognitive neural networks, which is a fascinating way to understand human rationality and says a lot that I think needs very deep thinking through. What I want to know then is, how is it that we are not the Borg, if that accounts?
How we are not what?
The Borg in Star Trek, where they are all identical robots, that they are one mind and they are one entity. And the answer to that has to be because of these things that we call mutations, that there is something that distinguishes one human being from another human being.
There are many things that distinguish one human being from another human being, and they're not just in the DNA sequence.
Absolutely.
So where the position in the womb is one thing. I mean, you look at twins who have most of their DNA is identical, and they're very, very different. So it's clear that.
But the reason why I draw attention to that particular one is I wonder if the work of moving to neural networks, as opposed to rationality as belonging to some Cartesian ego, cogito, or individuals who have IQs, etc. If we make the move from that to the system of neural networks, I wonder if that actually gets us where we want to get here. Because I take it that something you're really resisting in your analysis is the reductive account in which we just become our DNA as these materialistic bodily elements, that in fact we are so much richer as human beings than that. So I was wondering if you could speak to how the move to the system allows us to get past that reductive materialistic account, because it still doesn't allow us to make the connection between the human genome and rationality. And I think rationality is really somewhere where we need to go if we want to talk about human nature. And I just say that as a philosopher idealist who hopes that there is such a thing as rationality in human nature. Thank you.
That's it. Wait a minute, don't go away. That's a mouthful. Let me try to pick up on a couple of things. First, the Foucault, because I'm not going to remember all the things. Our conventions about disease, what we identify as disease and what we tolerate in the range of normality are there's of course enormous amount of feedback between what we identify as mental illness, for example, and what we conceptualize as normal. But prior to that, I'm referring to a prior step. We cannot talk about abnormal. For example, an organism that dies at the age of 30, let's take a solitary human being, dies at the age of 30. Is that normal? Or is that a disease? I mean, there's no formulating of the question in isolation. It's not a property of an individual human being.
I guess, I'm trying to reject normal and abnormal, because I research in Africa where it's actually perfectly normal, as it were, for children to die at age 2 of malaria, where life expectancy doesn't go past 40. So actually, I'm not clear how we understand normal here, but if we get rid of the notion of normal and abnormal, we end up with the idea that mutations are simply a source of diversity, and it's human diversity that makes us not into the Borg, and that must be a good thing. It's just that some mutations are more functionally useful than others, and the ones that aren't functionally useful end up getting us killed off earlier than we might have expected.
I think your example of the children in Africa is actually making my point, that it's not abnormal to die at the age of 2, precisely because in that environment the norm of the community is to die at very young ages. Now I've forgotten your other questions.
Well, just about how whether neural networks actually get us out of
neural
the
networks,
reductive
and what is the system?
materialistic.
Right. This, not just one systemic relations are, you have to decide on what level you're defining your system. So neural networks, neural networks are inside the brain, neurons are inside the brain, so you don't have neural networks. Well, the usual thinking about neural networks doesn't touch what goes on outside the brain. What Merlin Donald is primarily interested is in the cultural networks that connect brains. Embedding humans in, a human being is a system. The cell is a system. The organ is a system. The body is a system. The community is a system. The ecology is a system. These are different levels of systems, and they involve different levels of networks and feedback.
I had a couple of questions. I think they're both short and discrete. One was, because I know you're very interested in relationality as well, and how, and you've mentioned in conversation that we're not the only animal that is very social and developmentally constituted relationally.
We heard about quorum sensing in bacteria.
Yeah, and so there's, and I'm sure you're aware of this. There are a number of ethical traditions associated with religious traditions like Buddhism, where they see the interconnectivity that you've talked about as the ground for a certain compassionate response to the good life, that avoids some of the questions of moral philosophy in the so-called Western canon that have started from questions of, well, why be moral? And it starts with universalized self-interest and gets into a lot of bogged down, convoluted discussions of how to actually motivate someone who is fundamentally selfish. In this context, where you're seeing interdependence and relationality of the kind you're talking about, including distributed cognition, it takes away the self in the sense of isolation and puts us in a whole new space of relationality. And I just wondered if you could speak to that. And the second one had to do with your comment about bacteria in humans, and how if you wanted to actually have a look at the DNA substrate, you'd have to include bacteria. And one of my worries about the talk about human nature versus non-human nature is it still allows for this gulf between the human and the non-human in a way that is taken down a bit by the talk of bacteria. But I'm also wondering, as you think of us as relational beings and the developmental trajectory of, say, me from when I was a wee one to where I am now, a number of non-humans played integral parts in my developmental trajectory. And so it's not just fellow humans that have actually shaped me. And it continues to be the case that there are a number of non-humans in my life that continue to shape me and who I am and what I am. And I don't think I'm unique in this. And I wondered if you could speak to that as well.
I quoted John Donne, "No man is an island." There are no real islands. There are no closed systems of interest in the world. And the question is one of attention. Where are you focusing your attention, and how, and what kinds of distinguishing characteristics are you going to point to? So no man is an island, but we still speak of individual men. Yes, of course, animals and animal interactions are obviously very important, more important to some people, not to me. I grew up without them, but there are all sorts of interactions. Computer interactions, for example, are tremendously important in the shaping of the brain, in the shaping of the human.
Can you speak to the ethical implications of your discussion of relationality? No, you're not going to touch
I'm going to pass on that.
Okay.
You mentioned that it's not just the genetic sequence, it's also the environment, which I thought was fascinating. So my question, well, and also that we can change our own environment. You mentioned writing and computers. I was wondering, do you think that we can change our own environment to the point where we cannot evolve to adapt to it, and so, or we can.
We can change. Can we change our environment in ways that we cannot adapt to?
Yes, yeah, that's what I mean.
Absolutely. Think
Okay.
about global warming.
That's exactly what I was thinking of. All right, thank you.
There you go.
Hi.
Hi.
I was just wondering about a little question about scientific funding. Since I'd heard more recently, I thought it was interesting. There's you usually hear with things like cancer research, you'll hear about these huge, companies and grants that are working towards finding the cure for cancer, which sounds a lot better than, say, finding a variety of particular tests to several different 100 types of cancer. It sounds much more promising, and so it's easier to give funding to those types of groups. Do you think that in the type of shift that you're describing in our thinking towards a sort of away from this sort of genetic holicism to a kind of I guess more a more abstract idea of human beings as not only studying their DNA or the genome, but also the DNA codes and all of these organisms that live on us and that sort of thing. Do you think that there's going to be funding difficulties, or that it might be more seductive to keep this genome thing going, at least just for the funding? Or
No.
And it's clearly a bad thing to do that, but do you think
No.
that there's implications that way?
It's not that it's more abstract. It's just it's a different focus for the research. So even you talked about the cancer, the mission to cure cancer. Our thinking about cancer itself has been transformed by these developments, and the kinds of research that people are now doing in trying to understand cancer go way beyond DNA sequences and search for BRCA genes or whatever. It's the cellular context, all kinds of the epigenetic changes that are extremely important in cancer. So it's not that in fact, the funding itself has shifted from genomics to proteomics to epigenomics to, you name it. I mean, the money has followed this shift in mindset.
So I came away, great lecture by the way, great talk.
Thank you.
I came away with the notion that as research progresses, there's very little unique about the human being on a fundamental level, and that's being progressed.
Is that what you thought I said?
No, that's my interpretation. And as you look at research delving into ethology and neuroplasticity and genetic sequences and regulatory networks, these things are present in a variety of different organisms, most animal species, if not all.
We're much more plastic than most animal species. I mean, we are unique. I mean, don't get me wrong.
No.
It's just that our genomes are not that different.
I agree. Yeah. But also in terms of plasticity as well, to a degree. There's a difference in degree.
Right.
But on a fundamental level, these things are present in animal species as well. So my question is now that is the issue of human nature perhaps becoming a little bit more irrelevant, and should this be focused instead on an issue of speciation and identification of differences that define species rather than identifying fundamentals in human nature?
Question has shifted, and I'm not sure how meaningful the search for human nature in whatever terms continues to be. I'm sorry, I just lost the thread. Say the last thing you said again.
Should it be thought of as an issue of speciation rather than of human nature?
We all have our blind spots. Things that, I myself am a little technophobic. In spite of my degree in physics, there are some things I just can't seem to get myself to think about, and speciation is one of them. So, sorry.
Thank you, by the way. I want to give a few thanks before I give a thanks directly to Dr. Keller for pulling this series together, and especially this talk. It's been a series running across Canada with different lectures in different cities, all of which you can see eventually up on the web at the Canadian Centre for Ethics and Public Affairs website. But I'd like to thank the University of King's College for providing the facilities for doing this and the most vibrant intellectual atmosphere in the country, easily. For this, and this is absolutely true, we knock 'em dead here. For this lecture, Genome Canada and Genome Atlantic have supported both parts of the lecture series and especially this lecture, and we'd like to thank them. The Evolution Studies Group in Halifax, vibrant group of scholars that are interested in all aspects of evolution, cultural, historical, biological, and personal. There's, of course, the Situating Science a knowledge cluster, of which I have something to do with. I can't remember the name of it. The Canadian Institute for Advanced Studies, which has also supported this lecture series and this lecture. And of course, my colleagues in the Canadian Centre for Ethics in Public Affairs. And there's two people I want to really point out. I know that you don't even know them probably, but they really are very important for making this thing happen. Andrew Fenton, who has worked like a dog and has disappeared somewhere to go work like a dog somewhere, and pulling this together, and there he is. Give us a wave. Hi, Andrew. Who is a neuroethicist and about to return to his colleagues and their brains over at the Novell Tech. And Chris Stover, my colleague at the Canadian Centre for Ethics and Public Affairs, who is unbelievable at pulling national things like this together and has more things planned. So please do look at their website and our website for future activities in trying to understand our place in this tangled bank. Thank you very much. And thank you, Evelyn Fox Keller.